DNA fingerprinting test. So DNA fingerprinting test is developed by the Oleg Joffrey. The fingerprinting types may be ordinary fingerprints and genetic fingerprints and also DNA fingerprints. In case of the fingerprints, by taking the prints of any individual persons, we can identify the unique persons, and that is also used in case of the Aadhaar cards. In addition to that, we have the genetic fingerprints. In case of the genetic fingerprints, it can be used in case of the blood groups. The blood groups which are there in the form of A group, B group, AB group and O group, based on the multiple alleles. If a child is born for the parents, what is the blood group likely to occur to the child can be decided by taking the genetic fingerprints. So for example, A group is there and the O group is there. In this, the multiple alleles may be in the form of IAA and IAO type, and in case of the O group, in the form of IOO like structures. It is possible either in the form of the if it is homozygous IAA, in this case only it is possible to come in the form of the child in the form of the blood group A. If it is in the heterozygous condition IA and IO like structure in A group, it is possible to come either A group or the O group. That is also useful in the genetic fingerprints. The latest DNA-based fingerprints developed by the Oleg Joffrey. So by using the satellite DNA in the form of the VNTRS and the STRS. So the VNTRs, variable nucleotide tandem repeats, they are highly repetitive DNA molecules which are there in the genomes, and they are having the polymorphism. With that polymorphism, we can detect any person based on their fingerprints. In this case, we can see that by digesting the restriction enzymes, whatever the VNTRs are there, they will be done southern blotting, and by using just certain probes we can identify these bands. So suppose a child is born to the respective family is decided by taking the variable nucleotide tandem repeats from mother and father and child. We can see the bands, 50% of the bands will coming from the mother and another 50% of the bands will coming from the father. Based on that, we can see these bands in the form of the variable nucleotide tandem repeats by doing the southern, and we can do the blotting technique, and we can identify these bands with the respective probes. With this, we can take the X-ray film and identify the child is born to the so-and-so father. So based on that, we can do it. So whatever the VNTRs are replaced with simple tandem repeats, STRs. So in case of the VNTRs, which are forming in the human genome by unequal crosses. In case of the simple tandem repeats, these STRs are forming in the form of the 5 to 10 nucleotides length, and while DNA duplication is taking place, the DNA polymerase slips out from the template and again it binds to the DNA template. So because of that, these simple tandems repeats are developing in the human genome. These simple tandem repeats are millions of copies are there human genome. So recently, the Federal Bureau of Investigation of the United States developed the STRs by 13 STRs from the human genome that is used in addition to XY chromosomes. So these STRs will be multiplied by using the PCR technique by using the fluorescent dyes. So with this we can identify any person based on the simple tandem repeats, and also it is useful to maternity test. And also in hospitals we can need, whenever the child is born to respective mother, also it is tested maternal test. So the child is born belongs to the which mother, or a child is belongs to which father, paternity test. And also we can detect the crime region wherever the crime is occurs, any murder or any other cases are occur. We can collect the blood samples and also hair follicles, and we can do the PCR, and we can do the southern blotting, and we can identify the respective person with the help of these DNA bands. So DNA fingerprinting is a rapid and a reliable test to identify any individual based on these bands in the form of the VNTRS and the STRS, simple tandem repeats, and this locus will be very easily identified by using the PCR technique, by using the fluorescent dyes. These DNA fingerprints not only to identify the humans, they are also useful to identify the different plant species and also the genome, any other animal species. Like this, this is one of the molecular based identification of the any individual rather than the phenotypic other markers. This is what DNA fingerprinting test.